welcome to this Brochure Foundation Meetup. I'm Sally Davies, editor at Eon magazine, and it's my delight to host this discussion about the ethics of organoids. We have today, as part of the dialogue, Professor Alison R. Mawatri, who runs the Mawatri Lab at the Department of Pediatrics and Cellular Medicine at the University of California, San Diego, and Professor Jan Helge Solbach at the Centre for Medical Ethics at the University of Oslo. So grown from human stem cells, organoids are clusters of living cells that resemble parts of the human body, from intestines to livers, kidneys to brains. These ingenious proto-organs reenact the way biological structures develop in the womb and represent a major step forward in biomedical research. So today we're going to discuss the major scientific and clinical frontiers that organoid research allows us to probe, and also some of the philosophical challenges that they raise. But before we dive into the substance of the discussion, I thought I'd throw open the floor to our researchers to talk a little bit more about their personal journey to this area of research and what catalyzed their interest in organoids. Alison, would you like to start?
Yeah, sure. So I'm a geneticist, and from the beginning, I was interested on brain evolution as well as neurological disorders, especially diseases that affect human socialization. So this is what really distinguishes us from other species. And conditions where the social brain fails, such as autism or Williams syndromes, affect millions of people in the world. So I thought that I could contribute by providing them a better care, better treatment. So we turned into models of diseases to try to understand and come with new treatments. And most of the biomedical literature, it's focused on animal models, mostly mouse, and we fail miserable to find cures or treatments for these neurological disorders. So I started getting into the field of stem cells and brain organoids to create a human model that would be perhaps more relevant or complementary to the animal models, so we can find cures and treatments for neurological conditions. Of course, I mean, it's not only related to the brain. Organoids can also be derived from, can be created for other tissues as well. So we are helping like many people with diseases in all kind of tissues. So that's why I turned into brain organoids.
Sorry, I just realized I hadn't unmuted myself. Jan, and what's been your journey towards studying organoids? Where did your interest come from?
Well, for many years, I have been fascinated by this gray zone area between person and things. And in fact, it started when I was coordinating a European project on the ethics of research biobanking. And I was fascinated. Why the heck do we call these entities biobanks? Why do we use the bank metaphor? Are there other metaphors we could use instead? And we then tried out different analogies. Suppose we made the analogy to libraries or to art collections or to military conscription or tax obligations. How should we then think about these collections? And that brought us also to some striking things. When we regulate, for example, medical research, we regulate from the vantage point that was introduced in Roman law many hundred years ago. That is there is a categorical distinction between persons and things. Either entities are things or they are persons. And the fascinating thing with organoids is that they don't fit neither in the box of persons nor in the box of things. And just let me illustrate this. For example, some also scientists, they say we construct these models, these organoids, or we build 3D cultures, or we create. But maybe the right metaphor would be we nudge cells to assemble and make these organized structures, which are less complex than organs, but more complex than cells. So it's a kind of nudging.
That's a fascinating way of thinking about it.
It is not sheer construction, because these cells have some inner capabilities, and we stimulate, or the researchers, the real hardcore researchers as Alison, stimulate these cells to form these more organized 3D cultures.
That's really helpful, Jan. Well, I think before we launch into the kind of conceptual frameworks that we might use to understand organoids, I think it would be useful just to get some pieces of the puzzle on the table in terms of the main scientific advances, the really exciting areas of research. Alison, perhaps you could speak to that about what kind of discoveries and possibilities has organoid research unearthed that means it's got some runs on the board. What sort of innovations has it led to so far, clinical or otherwise?
Let me start with the technology per se. And the technology of organoids or creating these three-dimensional structures exists for a long time. And for a long time, they were, I would say, passive, inert. I mean, the cells, as Jan pointed out, the cells do it by themselves. More recently, we are optimizing protocols to make the cells do something that they were not supposed to do, or they're not programmed to do. So that's what I call like a more active research. Also, what's happening in the past years is that these organoids become more functional. And functional, I mean that the cells are expressing something that is very similar to what they do in the tissue, in the organism. For example, a pancreatic organoid is now expressing insulin. So in the past years, you have like a pancreas organoid, but there is no insulin. It's still useful, but there is no insulin. So recently, in my area for the brain organoids, we had the same thing. I mean, we had blobs of cells that resemble the human brain tissue, but they were not functional, meaning that the neurons were there, they were not forming connections. It's still useful, but we're not quite there yet. Then, I mean, suddenly we figured out a way to actually induce these neurons not only to communicate with each other, but also to form complex networks. So they got and start forming what we call neural oscillations. That is exactly the same thing that you can get by placing electrodes in your brain. This is what we call EEG. So before these organoids become functional, we could learn something about the molecular level, about the cellular level. I'll give you like a quick example. We show the causation between the Zika virus and the outbreak of microcephaly in Brazil in 2016. We couldn't do that with mouse models. We couldn't do that with any animal model. So it was the organoid who actually shows the causation. So we could attack the virus and not something else in the environment. And this was simply by exposing the organoid to the Zika virus. And we saw that the Zika virus kills the cells and reduces the cortical plate, so mimicking the disease in a dish. That was very clear and allow us to move forward. Now that we have functional organoids, we can get to diseases that are not related to malformations, but diseases that are related to network formations, such as autism, schizophrenia, dementia. So these are all conditions that affect how the neurons communicate to each other. So that's why I think by creating better models, by increasing the sophistication of the organoids, we are getting closer and closer to the real tissue, to the human brain. And of course, that brings some ethical considerations as well.
And how live do you think the ethical considerations are, Alison, in terms of, there seems to be a trade-off. On the one hand, the more sophisticated the organoid becomes, the more useful it is for research purposes, but the greater the risk that we could see a brain organoid displaying signs of consciousness potentially. Do you think that that's something that's a real risk? And if so, how are you offsetting that risk? How would we know if a brain organoid were conscious?
That's a great question. I think it's inevitable. We're going to do it. It's going to get there. And we have a reason to do that. As I said, we want to help millions of people with neurological conditions. So we're going to get there. I don't think we are there yet. I don't think the organoids that we have any evidence for a consciousness activity. But we are proposing to experimentally validate some of the evidence that these organoids might have conscious activity. I can give you like 2 experimental evidence. I mean, and by the way, just to clarify, there are many consciousness theories. So we have to use the ones that allow our model to be the experiment, because there are consciousness theories that suggest that the consciousness is outside the body, is outside the brain. So then there is nothing we can do. We will never know. But there are specific theories that suggest, for example, that consciousness arising within, and there are specific characteristics of the brain that we can check. One example is the exposure to anesthetics. So if you anesthetize a brain, a human brain, all these neural oscillations goes away. Would the organoids do the same now that they have neural oscillations? If we treated them with anesthetic, will these oscillations goes away? So that's a question. The other one is what we call the zap and zip. It is something that you challenge these organoids with a magnetic pulse, and then you measure the complex of the reverberation of that stimulus. And if you are conscious, you have certain index, and if you're not conscious or have a brain damage or under anesthesia, your index is quite low. And then we can place organoid in this between and see, I mean, how conscious or not conscious are these organoids. So these are the type of experiments that we are proposing as a checkpoint on the technology.
Fascinating. Jan, I'd like to bring you in here. Do you think this distinction between conscious and not conscious organoids, does that map neatly onto the distinction that you raised earlier between persons and things, or are they somewhat at an angle to one another, or some sort of overlapping Venn diagram?
Let me try to unpack that by what is driving, for example, organoid research is the translation problem. How to translate knowledge generated to alleviate disease-induced suffering from animals to human beings. And there is a huge gap. And how can we close that gap or make this gap smaller? And for this reason, organoid research is such a fascinating new or fairly new research field. I'm coordinating now a big European EU-funded project on these issues, the ethical and regulatory challenges. But I think we need to also communicate very clearly. First, if you look at publications within organoid research, the biggest field still is gut organoids. And in a way that complies well with the fact that the gut is the biggest organ in the body. And here is a lot of research going on. But also, when we talk about medical and health related research, we divide between phase 1, phase 2, and phase 3 clinical trials. And you can correct me, Alison, but probably 99.9% of all organoid research today is a kind of research between laboratory research and phase 1 trials. So we are still far from entering the clinical room and treating patients for any kind of disease. But let me add to that. I'm not so concerned about will the researchers, as Alison, be able to create organoids, brain organoids that are so complex that they will have some capabilities in the direction of some kind of consciousness. And the reason I say that is, well, maybe it is a bit silly to say, but the French philosopher Alain Badiou say, human beings are animals who like to go to the cinema. So why should we continue to be so afraid about the boundary between animals and humans, if humans also are animals? Immanuel Kant, he was strongly against medical research using research on and with animals. Not because he was concerned about animals, but he found that this would be an affront against the dignity of human beings. And I think Kant was absolutely wrong. And I don't think the real ethical issues in organoid research pertain to brain organoids. Let me draw attention to another ethical challenge. There are good reasons to believe that organoid research will, in the near future, generate a lot of patent applications. These organoids have been nudged from cells that derives from identifiable citizens on this planet. And that raises the question, how should one share or distribute the economic incomes of this extremely fascinating research? Should everything go to the researchers and their institutions, or should something be fed back to the donors? That is an ethical question that relates to all kinds of organoid research.
I mean, if I can jump in there, it's also a pragmatic question insofar as intellectual property as a concept was created in order to spur innovation. So the idea being here that if the researchers were permitted to keep the proceeds of their innovations, it would lead to more and better organoid research. That may or may not be the case. Presumably there are other incentives we could create. My question here too is that, are there any precedents for this? I mean, in terms of how we might regulate organoids, I'm wondering about whether something like the global response to recombinant DNA technology, I mean, are there precedents for biomedical regulation that have worked well that we feel could be translated to the organoid domain, or is it so sui generis that it requires a categorically different approach?
No, let me go back to this Hybrida project I'm coordinating now. This is a kind of project that the EU Commission fund. It is called SWAFS project, Science with and for Society. And the Commission want us to increase a genuine dialogue between the research community and the community at large, not just to inform the research, the community at large, but also it's a kind of acknowledgement that generation of new knowledge by researchers is not solely an activity generated by researchers. We need also feedback from the community at large. And I think that is particularly applicable to this field. Well, in organoid research, if you read scientific papers, you come up with concepts such as hybrids and chimeric entities. Well, these are ancient concepts coming from different mythologies. And some of these most famous chimeric entities are fire-spitting monsters, which are perceived as extremely dangerous. But we should also keep in mind the fascinating thing that one of these ancient Greek chimeric entities is considered, Chiron, is considered the father of the god of Western medicine, the father of, not the father, the teacher of Asclepius. So you have these also, you have this double perception of chimeric entities, and certainly some of the concerns, for example, how if you introduce human brain cells into mice, are you then humanizing mice? Or if you are introducing mice brain cells into humans, are you then micellizing humans? Well, the background for these concerns relates to these ancient concepts and metaphors that, interestingly enough, even scientists, hardcore organoid researchers are using today. But not all researchers are aware of that these are not scientific concepts. These are metaphors that try to capture something.
So if I understand you correctly, Jan, what you're saying is that in discussing organoids, we're invoking some of these very ancient and long-standing tropes that speak to cultural understandings that we possess about the appropriate relationship to our bodies, other bodies. I'm thinking here too of the Freudian notion of the uncanny and this idea that something that is sort of almost but not quite human that we find very disturbing. And so your claim is that we need to be attentive to the work that these concepts are doing in terms of how they drive regulation, indeed possibly how they drive research. Would you agree with that, Alison?
Yeah, because no, sorry if I interrupt. I think the key challenge also in this field and in all kinds of medical and health related research is trust. For example, now during this pandemic, there is an epidemic that goes on along with this pandemic, and that is an infodemic, and the distrust, a growing distrust. And I think to build trust within this new fascinating field, it is so important to engage with the society at large. Researchers have to move out from time to time, maybe from Friday afternoon to Monday morning, out of the laboratories and engage with people.
Well, I know for a fact, having worked with Alison at our magazine, that Alison takes his responsibilities when it comes to public engagement very seriously indeed. Alison, I'd like to come back to, I'd be very interested to hear your thoughts about the relationship of organoid research to some of these ancient metaphors. I'd also be curious to hear your thoughts about what, as one of the world's leading experimentalists in this area, what you think, what your reflections are about an ownership structure for the innovations and benefits from organoid research.
Okay, yeah, let me start with the first questions, and I totally agree. Yes, I think researchers end up using those terms out of creativity because we can't figure out a better term or give a new scientific term, so kind of borrow from place to place. There is also the contribution of media in an effort to kind of simplify the research, end up using terms that are out there already so people can connect better. And one example in my field is the term mini brain. So it was coined to suggest that these brain organoids are miniaturized human brains, which is not, it's not true. But it's something that people directly connected with that. So we pay a price for that. And the price is the concerns that people have for the research. So this is something that we always have to be aware, and it's a balance, how to communicate science in a way that is simple, it's easy, it's digestive, even very complex research without falling in the traps that are easy to fall by borrowing those concepts from other fields. So this is, I mean, we have to evolve. We need like more science education, including for scientists, to kind of come with better terms. Regarding the ownership, I do agree that in the ideal world, some of the revenues of whatever research should go back to the subjects. But just remind people that also the biomedical benefit is on the line as well. So if nobody contributes to the research, there is no benefit at all. So sometimes, I mean, the weight is that would you contribute in having like a treatment or a cure for your disease in the future, even if you have to pay for that? So again, in the ideal world, I think the person that contributes should not have to pay for the cure. But unfortunately, that's not how it's done right now. If there is something that could go back to the donors, I'm 100% on board.
And as it stands currently, are there any restrictions on what sort of patents or intellectual property you can place on organoids? I mean, would it be possible for me to give up some of my stem cells and for an organoid to be created from my body, and then for you, Alison, to patent that organoid and I'd have no say whatsoever over what happened to it.
Yeah, that's exactly how it's done. So before I consent someone for my research, they have to understand and sign a consent form. And it's written there that you are donating yourselves, you're going to make organoids, we're going to find a cure for whatever disease we are studying. It might be that this cure, this treatment will be commercialized and someone will make tons of money and you're going to not receive anything. So you have to donate out of, you have to be selfishless, let's put this way, to donate the cells for the research. There are instances where we can pay something back to the donor, but at these very early stages where the science is done in a preclinical setting, because most of the research that we do fail, this is also important, 99% of what we do fail. So most likely, I mean, that research that I'm working will not lead to a medicine or a revenue down the line, but eventually 1 in a million will, and that's what we're talking about. That's why, I mean, it's important to have the contribution and understanding of the public. Otherwise, I mean, we make no advance. There are even, I mean, people, sometimes, I mean, we seek relatives or even people in the lab to contribute to the research, to be subjects of the research, because sometimes it's hard to attract people, especially when it requests like an invasive technology, like a skin biopsy or a little bit of blood. So you actually have to go to the lab and be subject to that. And you can imagine how to do that with an autistic individual, for example. It's really hard.
It's interesting. It seems like, I mean, maybe this is somewhat reductive.
Sorry, Sally. I have to leave and enter again because there is a lot of echo. So I come back in 1 second.
Sounds good. Let's see if there's less echo now. It seems like there is. You know, I mean, it does sort of strike me, Alison, speaking about this. I mean, it reminds me, of course, of the Henrietta Lacks story and the HeLa cell line. And this is the scenario, you know, writ large, that we want to avoid. And of course, you know, I'm conscious of Jan's note of caution about not getting carried away at the prospect of conscious organoids, but it does seem, you know, it does seem slightly daunting to think, particularly when it comes to neural organoids, the idea that a kind of a copy of your brain could potentially be created that you would have no control over. It's interesting too, actually, that you know, to some extent, I think the conversation around neural organoids sort of mirrors some of the conversations around artificial intelligence, where we focus on these kind of quite long run questions about will we create an artificially intelligent creature, when actually the ethical concerns, the day-to-day ethical concerns about what algorithms are doing to our daily lives are kind of far more pressing. And, you know, potentially these concerns around consent when it comes to the donation of tissue and the implications of the research are kind of much more pressing than the question of what would we do if a brain organoid became conscious.
Yeah. And I'm afraid that the science moves faster than the kind of discussions that we are having. And that's why, I mean, I thank you guys for putting this together, because the more we talk about it, the better.
And what are some of the, I mean, we sort of touched on this at the start of the conversation, but what are some of the, have you got any emergent results that you can speak about from some of your latest research findings?
I'm assuming you are asking about the consciousness, any evidence of consciousness in these organoids?
Well, we could talk about that. That would be fascinating. But you know, it would also be interesting to hear whether you'd made any advances in your study about the origins of autism and schizophrenia, for example.
Yeah. For the consciousness, we are still creating like setups for these organoids. There are lots of technical challenges. The tissue is not as hard as, for example, the human brain. So that has lots of consequences when we are trying to measure and record activity. So these are experimental challenges that we have to go along the way or fix along the way, because we never worked with those tissues before, so we're learning. From the basic science, yes, it's moving quite fast. We are learning, especially on autism, how different genetic perturbations can actually change the path of neurodevelopment. and affect how the networks evolve to generate a behavior later in life that would be similar to an antisocial event that someone might have it. And we are also using these organoids to try to probe what kind of environment makes these organoids more susceptible in combination with these genetic variations to create the autistic behavior. So there is a gap between the cellular molecular analysis that we can do with the organoids and the human behavior, but nonetheless, I think we are doing the first steps to bridge that gap.
Fascinating. Oh, Jan, I've just noticed you're back now. I hadn't seen you before. Did you want to jump in?
I think it's important, although I maybe threw some cold water into the discussion. Probably the most promising part of organoid research when we talk about clinical application relates to drug testing. That, one of the big problems with drugs is that drugs have been developed based on randomized clinical trials, including hundreds, if not thousands of patients. And that means that the results are average results. But we know that every patient with the same disease might react differently to the same drug. And this is a huge problem, how to develop drugs that are more precise in terms of positive effects and having less side effects. So it is this vision that maybe organoid research will be a better way of future drug testing to develop safer and more precise drugs. And certainly the one vision that has become more vivid because of organoid research will it be possible in the future to develop real personalized medicine? And these are important visions, and I think when we talk about where clinical applications will come first, probably it will be in relation to a new way of testing drugs.
So that's fascinating, Jan. I mean, what I hear you to be saying, and this is actually kind of picking up on Alison's last point about some of the frontiers that his lab is probing at the moment, is what organoid research allows us to do is sort of capture the complex causality of a lot of diseases, that we move away from this kind of simplistic model of the human body as a kind of machine that gets broken and then you intervene with a single fix. And instead we understand there's this very complex genetic, environmental interplay that produces certain path dependencies about how diseases evolve, and that we're better able to capture that causal picture if we have a kind of functioning proto-organ compared with when we have a human population from which we can only discern kind of statistical generalizations. Is that a sort of fair description of why organoid research is so helpful in this area?
Yeah, but there is also a huge challenge to use a concept introduced by one of my colleagues, Henrik Vogt, the precision paradox. The vision of personalized medicine is to develop treatment options, drugs that are more precise in individual patients with less side effects. But the challenge is when you reach a smaller and smaller number to test, you can't use statistical methods. So the paradox is that before you can realize real personalized medicine, you have to cross a bridge which is filled with much more uncertainty and imprecision than present treatment options and drugs that have been developed based on randomized controlled trials. So you have this precision paradox. Maybe organoids will be one way that this paradox can be resolved or unpacked. Maybe.
What's your view on that, Alison, about the potential for organoids to bring us closer to personalized medicine?
I'm with you, and I'll tell you one experiment that is going on in the lab right now. So this is a clinical trial for autism, and we are studying severe autism. These are individuals that cannot talk, they're very aggressive, they have extreme antisocial behavior. So it's very the end of the spectrum, the more severe cases. And we are testing a drug for them. So there are 30 subjects on this clinical trial. For each one of the subjects, we are taking a little bit of their skins and from their families as well, and creating organoids, and we're gonna generate these neural oscillations for each one of them. And then we're gonna test the same drug that we are testing in the clinical trial in the organoids. And for the patients, we are also measuring their EEGs. So we'll have the same electrical readouts from their brains before and after the treatment, as well as for each one of their of the respective organoids before and after the drug. Our hope is that we're going to find a signal in these oscillations that might correlate with their more adult brains to see if they are responding to the drug or not. So we are putting the organoids to the test. Even though they are very simplistic, they're very reductionist in nature, they might contain something that can correlate directly to the brain of the person that they were derived from. So if that's the case, then yes, then maybe what we should be doing is more precision medicine, or creating clinical trials in vitro to figure out who's going to respond, and then testing in vivo. So that would save millions of dollars if we do that. So that's the experiment that's ongoing. We hope to have results by next year.
That is fascinating.
But that brings me back to another thing we did when we prepared this Hybrida project. In order to really address the ethical challenges in this domain, we need to address two other levels of uncertainty. First, what we philosophers call ontological uncertainty. How should we think and conceive of these entities? And second, epistemological uncertainty. How can we be sure that the kind of knowledge we try to generate is certain knowledge when we do not have statistical methods? We do not have clear theories of causation, etc. So there are uncertainties on three different levels. On the what is this thing or entity level, how should we understand knowledge generated from research on such entities, and what are the ethical challenges generated by these entities, by this new kind of knowledge generation. And we have to see these three dimensions at the same time.
I mean, it seems to me that part of the challenge here is a sort of meta challenge, which is it's not just answering these questions, but it's also answering these questions while still allowing the research to go on, because of course it's not that we can sort of sit and stop and wait for this to be bottomed out and tell Alison that he has to, you know, pull up sticks and wait for the word until the philosophers have resolved the metaphysical status of organoids. But then, of course, figuring out the appropriate pace of this research and making sure that the regulation and the ethical considerations are kind of that they're sort of woven into the research. I mean, that seems to be a very difficult challenge. We do have a question here actually from an audience member, Morel Gross, who's asked you, Jan, if you would speak to some of the proposed solutions that your task force might be exploring for this challenge you highlighted around the ethical distribution of the benefits of organoid research. Are there any kind of innovative distribution structures that your task force is exploring?
Well, first, we have a work package that will engage systematically with different stakeholders and interest groups. Because I think also when it comes to the ethical challenges pertaining to organoid research, we need feedback from different stakeholders, not the least also members of civil society. But I think just to take a side view now to the present pandemic, in spite of the fact that the majority of funding for the vaccine developments have been paid by governments in the global north, the pharmaceutical industry still insists on the same patent rights. And I think that will cause an important discussion. Are the patent rights as they now are designed and practiced completely outdated? If we move in a direction where there is a growing perception that knowledge generation in science and research is a kind of the generation that is not just produced by individual researchers. It is through a communication and engagement with the rest of society. And if that is acknowledged, then also the question about how should the economic benefits from knowledge, this kind of knowledge generation, should this benefit be distributed? And I think this will also be a challenge in organoid research.
It's interesting too what you said earlier, Jan, in fact, by starting the discussion with this framing of organoids as cells that in themselves have a certain agency, and that Alison and others nudge towards taking on certain forms. I mean, it's what you seem to be saying there is that organoids are not just a product of Alison's genius plus the community that he's enmeshed in, but it's also there's an intrinsic capability within these cells that can be directed along certain paths. I'm not quite sure what the ethical implications of that would be, other than to say, I suppose, life itself is patrimony of everyone. But I suppose that furthers your argument that the benefits of this research need to be more widely distributed. Although I'm not sure whether Alison would agree. Perhaps you'd like to jump in here, Alison.
I definitely agree. I don't see a clear solution right now, but the concept is very attractive to me. I deal a lot with very rare genetic mutations. So these are, individuals that have a mutation in a gene that nobody else has. So it's very private, very unique. And that mutation might allow us to understand a little bit of a specific pathway to help other people with different mutations, different genes. So the benefit of studying that rare mutation might be bigger than that is single individual, but to a whole community. So I feel that, that private mutation that causes lots of pain and suffering for that individual might be the key for a cure or a treatment for lots of other people. How can we compensate that one? Especially the ones that carry those unique genomic information. I think these should be the ones that should receive something back.
Yeah. I think there is a great potential that there will be there will be also in future some Henrietta Lacks around the corner. And this future Henrietta Lacks should not be treated as the former Henrietta Lacks.
Yes, Jan, we were saying this a little earlier that one hopes that we're all alive to those ethical quandaries, but of course the question around consent in an area of research where we ourselves don't fully understand what the potential applications are, it becomes very fraught. Something that is optimistic though that I take away from what you've said is that the COVID pandemic, as devastating as it's been for so many people, has nonetheless provoked a public conversation about the appropriate distribution of bodily risks and the appropriate distribution of gains from biomedical research, and a kind of more sophisticated, widespread understanding than we perhaps had before. So your claim is that this might sort of catalyze a more productive conversation around the appropriate way to manage organoids.
I hope so.
Okay, it's always good to end on a positive note. We are coming up to our 45 minutes. There's much more that I wanted to ask and speak about because it's such a rich and fascinating area. Perhaps just to close, I'm conscious that we've obviously talked a lot about brain organoids, which is Alison's area of specialty. But are there any specific kind of concerns or opportunities in other areas of organoid research? Jan, you of course mentioned that gut organoids were a very active field of study. Are there other specific ethical concerns with respect to organoids beyond the brain?
Well, certainly, if you take up the global perspective and you look at the global burden of disease, one at least should ask the question, is this the best investment in medical and health related research when it comes to reducing the global burden of disease? But the same goes for the vision of personalized medicine. We know that this is a kind of future medicine that will benefit patients in the global north, hardly ever patients in the global south. So there is, but this is in a way, it's an ethical challenge that in a way is not just a challenge for organoid research, but for a lot of the highest high-tech medical research.
That's a very fair point. I mean, I think, Alison, you're in a very interesting position to speak to that, of course, being from Brazil originally and of course now working in the US. What's your view about the ethical justification for organoid research and how we balance that out against investments in undertreated tropical diseases, for example, being one of the best buys in public health?
Yeah, no, you're right. So I would love to see some kind of benefits going back to these underrepresented populations that we actually, I mean, sometimes we take advantage of their biodiversity, but we never pay it back. And that's how the system is set right now. So they don't have enough support to advance their own sciences, so they become colonized by the advances of a more sophisticated scientific achievements by paying for the royalties, the patents, and the treatments that will be developed from their materials. And I think this is a broad discussion. It involves like DNA from Indians, ancient populations. So there's lots of problems everywhere that we need to solve. I just would like to mention one thing that you also talked about, potential ethical implications in other tissues. And this is my honest opinion. I think that that's why I like the brain organoid discussion, because I think it touches people more than other tissues. I mean, we have heart transplant, we have liver transplant, we've done those things, and people seems okay with that, or at least most people seems okay with that. But if you ask somebody, would you do like a brain transplant, people would go crazy. And I think that's because right now our society values a lot the brain as a personification of the individual. And that's why we have the yuck factor when you say, oh, it's your brain organoid here. So people are uncomfortable with that. That might change in the future or might not. Or might be that, yes, there is a connection between you and that tissue. And that's important, and we cannot discriminate. But I do believe that if we are working with a gut, a pancreas, a liver organoid, the concerns are reduced, if non-existent.
Thank you both.
Alison, wouldn't it be very interesting for you to nudge brain organoids taking cells from Mr. Bolsonaro and Mr. Trump and find out what the heck went on in their brains?
I'm afraid the organoids will not survive.
With that zombie-like prospect, I regretfully will have to close out the discussion. We have had another question come in from one of the panelists, but unfortunately we're out of time. But thank you both to Jan and Alison for this extremely enriching discussion. And I do hope it can be continued in some other forum another time. So thank you both for your time, and thank you everybody and the Brochure Foundation for hosting this meetup.
Thank you, it was a pleasure.
Bye-bye everyone, thank you so much.